This message is to members of the 21st Pacific Science Congress. It is a great honour for me to be awarded one of the Shinkishi Hatai Medals for 2007, and I wish to thank Professor Kiyoshi Kawasaki, President of the Science Council of Japan, for this special honour. I'm very sorry I'm not able to be at the 21st Pacific Science Congress to accept in person. When I first started my work in Australia's Marine Division of CSIRO in the early 60s as a young postdoctoral fellow, very little was known about the microalgal populations that form the plant pastures of the sea. My chief, Dr. George Humphrey, gave me only one instruction: to find out what plant pigments are in the ocean so that we could develop a chemical method for determining the microalgal biomass, so much simpler than cell counts and taxonomy, and more accurate. In the years that followed, I've had the great privilege of studying these marine plant populations, especially the marine plant pigments and the marine chlorophylls, which I came to love and were always telling us new things. The phytoplankton populations around Australia's tropical, temperate and polar seas from research vessels with colleague Dr. Gustav Hallegraaff. Building up of our microalgal culture collections from less than 10 species to our over 800 strains which we have at the present time, as a bank of precious experimental material for local and international use. Assisting the Australian aquaculture industry, particularly farming the Pacific oyster, and transferring hygienic expertise from the laboratory to farms, which are now very successful, and this work was done with Dr. Christian Garland. We've also improved our microalgal taxonomic knowledge with Dr. Marette Vesk, and we've been working on distinguishing toxic from non-toxic species, which can form harmful blooms affecting fisheries, both farmed and wild species. This work has been done with Dr. Gustav Hallegraaff. Learning more about the mycospore and amino acids, which can protect marine plant and animals for damaging UV radiation. Another aspect has been helping to ground truth measurements of surface ocean colour from space by remote sensing techniques, giving us global ocean coverage of phytoplankton productivity, so important in today's concerns of global climate change. In 1997, supported by UNESCO and the Scientific Committee of Oceanic Research, we published an international book entitled Phytoplankton Pigments in Oceanography: Guidelines to Modern Methods. This work was done with colleagues Dr. Fauzi Mantoura and Simon Wright to assist scientists around the world to be more accurate in their measurement of pigment concentrations in the oceans. This book has been so successful that a new volume updating the methods is now in progress. May I conclude by thanking CSIRO for the wonderful privilege of working in this field of microalgal biology, which has advanced in coverage from molecular to global over the years. I hope future advances will assist in monitoring biological, genetic and environmental changes in the ocean's phytoplankton as a guide to understanding and protecting our oceans from environmental change. My grateful thanks are due to 3 Japanese colleagues who assisted me with various projects over the years. Professor Kazuo Shibata from the Tokugawa Institute for Biological Research in Tokyo worked with me on the spectroscopy of marine chlorophylls. We were both on a cruise in the mid-1960s in Australia's coral reef area in North Queensland, and this was a wonderful international expedition. Professor Miyachi, Marine Biotechnology Institute, which has a laboratory in Shimizu and also in Kamaishi. He was interested in aquaculture and microalgal culture. And more recently, Professor Yasushi Koyama, who was interested in marine chlorophyll seed preparations for applied use. Professor Koyama very kindly agreed to be here in Okinawa to accept the medal for me. Australian colleagues I've mentioned already include Dr. George Humphrey, Dr. Gustav Hallegraf, Dr. Moret Besk, Simon Wright, Fauzi Mantura, Jeannie Marie Leroy and Christian Garland, and SCORE and UNESCO and CSIRO for funding over the years. I would also like to thank my husband, marine population ecologist Dr. Andrew Heron, who always gave me wonderful support. And more recently, the Chief of the CSIRO Division of Marine and Atmospheric Research, Dr. Greg Ayres, has been giving me special support. It has been a wonderful life amongst the marine microalgae, and thank you all very much for your kind attention.